everyone, my name is Evana Jang, and today I will be talking about the world's leading cause of death, heart disease, and how scientists can utilize CRISPR-Cas9 gene editing to unlock the heart's potential to regenerate heart cells as a treatment strategy. First, I'd like to introduce myself a little bit. I am from Hong Kong and currently a rising senior at Miss Porter's School located in Farmington, Connecticut. I am the co-editor-in-chief of a STEM journal, a head of the Asian Student Alliance, a summer intern at the Integrated Medicine Institute, and a member of my school's varsity basketball, track, and cross-country team. My intern experience inspired my interest in osteopathy, and I'm also fascinated about orthopedics and oncology. Now let's begin. Heart disease is the leading cause of death globally, taking around 17.9 million lives per year. This one disease accounts for 13% of the world's total deaths. The deadly nature of heart disease is due to the fact that once heart damage occurs, it is irreversible, slowly taking away a once thriving organ's ability to function. Heart damage occurs due to a buildup of fats or cholesterol in the arteries, which lowers oxygenated blood flow to the heart. This causes heart tissues to die, resulting in the loss of billions of cardiac cells known as cardiomyocytes and a permanent scar. The scarring and loss of cardiomyocytes results in a weakened heart pumping system, giving way to congestive heart failure. The reason why heart damage is irreversible is because cardiomyocytes can't regenerate unlike other cells in the body. Thus, the heart can't heal itself once damage occurs, making it very deadly as the chances of a heart attack occurring significantly increases. Additionally, people affected have a quality of life that is significantly lowered. Symptoms of shortness of breath, fatigue, sleeping difficulties, depression, and chest pain are common, which limits their daily social and physical activities. This underscores a vital need for heart disease treatments. And current treatments include heart transplants. However, the demand of patients needing organ transplants is way too large for the supply, so new solutions need to be created. The solution I'm proposing involves looking for ways to unlock the human heart's ability to heal itself using genetic editing to induce cardiomyocyte proliferation. While human heart cells aren't able to self-regenerate, some animals, including zebrafish, have this potential. Zebrafish are able to regenerate their heart after amputation of up to 20% of their ventricle. Knowing this, I conducted in-depth research of cellular pathways within zebrafish cardiomyocytes that allow them to regenerate, in which I came upon 4 distinct pathways that are heavily researched. The 4 pathways are the TGF-beta, NF-kappa beta, Notch, and Hippo-YAP pathway. Previous studies found that these pathways are induced during the heart's regenerative stages, and when inhibited, leads to defective regeneration of heart muscle. Knowing the key pathways that allow for zebrafish heart regeneration, I propose to use CRISPR-Cas9 genetic editing to upregulate those pathways in a pig heart to see if it induces cardiomyocyte proliferation. The reason I chose a pig heart is because before we can test on humans, the safety of the treatment has to be assured. Pig hearts have the closest resemblance to a human heart, so it would show similar effects. This CRISPR system is composed of a guide RNA and a dCas9 enzyme. The difference between a dCas9 enzyme and a normal Cas9 enzyme is a dCas9 enzyme is able to upregulate gene expression without making double-stranded breaks, which makes it less harmful in case off-target effects do occur. The dCas9 protein will have an activation effector domain fused to it, which is what allows the gene expression to get upregulated. Essentially, the guide RNA leads the complex to the target gene, and the dCas9 protein will upregulate the gene. CRISPR, along with somatic cell nuclear transfer, will be utilized to create pigs with genetically modified heart cells. Researchers will use the CRISPR complex to upregulate a pig cell with the desired changes. Scientists will then use somatic cell nuclear transfer to generate pigs from the edited cells. This involves replacing the nucleus of pig eggs with the nucleus from a gene-edited pig cell. The eggs will then be transferred to surrogate sows to develop and grow until natural birth. The pigs born will be composed of cells containing genetic edits, and so their organs, including the heart, will express the characteristics of the upregulated genes. The 5 pigs, including a control with no genetic changes, will be grown in identical conditions to ensure the only variable is the type of upregulated genetic pathway. The pigs will be fed a high-fat diet and given minimal exercise to stimulate heart damage and a loss of cardiomyocytes. An MRI scan will be conducted monthly to analyze how the heart pumps blood throughout the pig's body. The screens will also visualize the number of cardiomyocytes within the hearts and compared with different months to see if there is an increase. Data will be collected for 18 months until the pigs reach full maturity. At the end of the 18 months, the MRI data from all 5 pigs will be compared to see which genetic pathway manipulation yielded the greatest restoration of heart function and whether there is any proliferation of cardiomyocytes. Research will follow all protocols of ethical animal testing. While the study involves inflicting heart damage on the pigs, it will be done so in the least invasive way through diet changes alone. To reduce any stress that comes with inducing heart damage, the pigs will be well cared for daily and offered opportunities of enrichment. At any point where the experiment causes the pigs pain, anesthesia or analgesic drugs will be administered safely to eliminate any suffering and distress. To conclude, it is truly vital to find a solution to heart disease, a condition that has taken the lives of many and severely impacts the quality of life of those who have it. With the availability of cutting-edge tools such as CRISPR-Cas9 and somatic cell nuclear transfer, it is up to us, people passionate about healthcare, to think out of the box for potential solutions that can one day be a reality. So far, much experimentation surrounding manipulating heart regeneration has solely been done within zebrafish, but it is time to move on and ethically experiment on a mammalian heart to step closer to implementing this solution into a human heart to treat heart disease. In this experiment, if any pathways show success in reversing the effects of heart damage, researchers can then move on to exploring how to upregulate those same genes in human hearts. By harnessing the power of cutting-edge gene editing, we could transform the future of heart disease treatment and offer millions a chance at a full, healthy life. Here are my references, and I want to thank GHLC at Johns Hopkins for giving me this opportunity to present, and thank you all so much for listening.